Hey everyone, this is the second section of our Unit 3 as we are going to continue talking about DNA, but this time we're going to talk about how DNA is copied as we make new genetic information. So it is done semi-conservatively. That means that half of every DNA strand is kept and a new strand of DNA is brought into that. What that means is that as DNA is copied, it's split in two, and new nucleotides are brought in to make full copies. So this diagram here kind of shows that. If you look at the dark blue, every iteration of copying the DNA, what process we call DNA replication, becomes 50% old DNA and 50% new DNA. So 2 generations later, that second copy, right? We've got a new brought in, and the cycle just kind of repeats itself. So we always keep the original strand present, but it's split up and new nucleotides are added to it. That process happens in 3 main steps: initiation, elongation, and termination, where that DNA helix is uncoiled. The chromosomes are spread out, so it's not condensed. The helix itself is separated and new nucleotides are added in. The new nucleotides are always matched to the existing nucleotides, and then they are brought back together so that those hydrogen bonds can form and hold the 2 halves of that ladder together. Then it's coiled back up and it's checked for errors. This is what that process looks like. I like this diagram as the topoisomerase is going to move ahead of what we call the replication fork, where the DNA is actually split in half. And it's split in half by helicase that breaks those hydrogen bonds, right? Then we've got this issue. Because DNA is directional, we're going to have what we call a leading strand and a lagging strand. The leading strand is the easy part. So DNA polymerase, which is an enzyme that adds nucleotides to the DNA, right, can come in right away and work off of the DNA that's there, that half of the DNA strand anyway, and it can add nucleotides in that pattern that we talked about when we were talking about Chargaff's rules: A to T, C to G, G to C, T to A. We're making DNA, so thymine is going to be there, guanine, cytosine, and adenine, all present. It's going to keep those same percentages that we talked about with Chargaff's rules because they're just matching it up. It's going to always move towards the replication fork as it moves towards that, and it's continually be opened up. The lagging strand, however, because the directionality goes the other direction, has to have a few things. All right. First is RNA primers attached via primase. Sometimes this is called DNA primase because it works on DNA, right? These primers just act as markers for the DNA polymerase to attach to, and they come in and does the same thing. It adds nucleotides the same way, but it's going to be traveling in the opposite direction because the 2 halves of DNA are anti-parallel. Because it's going to work in these chunks or segments, it leaves behind what we call Okazaki fragments. Those are named after the doctor who was studying them, and it is going to just show that the DNA polymerase has done its job, but it's leaving these chunks. Because as it reaches the material that already has had nucleotides added to it, it falls off. That means we have one last enzyme, DNA ligase. DNA ligase works kind of as glue, and it brings those sugar phosphate backbones together. It bonds them together to make the whole piece one unit instead of having those chunks, those fragments. So we've got the leading strand and lagging strand. For our question today, I want you to try to write a step-by-step explanation for the process. Based on how you took notes over that last minute and a half, 2 minutes, you might already have this in your notes, right? But I want you to specifically include the following enzymes: topoisomerase, helicase, primase, DNA polymerase, and DNA ligase. Technically, there are a couple DNA polymerases, but we're going to keep it simple and just call them all DNA polymerase. One will be on the leading strand, one will be on that lagging strand. But try to just set yourself up a step-by-step explanation for the process. It needs to be more in-depth than just initiation, elongation, termination. Right? This is how that genetic info is formed. It doesn't matter if our skin cells are replicating or if we are making sex cells for reproduction, right? Gametes. It doesn't matter. They're made the same way because DNA is always copied the same way. Later in the unit, we'll talk about what happens when that goes wrong, when we have mutations, and how those can arise and how they even can be fixed. But for now, we're going to talk about this more in class. I will see you there.